In this country and around the world, whose names we do not know, are not with us, is because the methodology that has governed science for the last century stinks. Louis Pasteur died in 1895, and it was not long after that that Max Planck, in the area of physics, explored some of the paradoxes in the behavior of light. This became known as the wave-particle duality, where we couldn't really figure out what was happening with these particles of light. And this idea was taken by certain people like Niels Bohr and others to mean there is no reality. Truth can never be known, that on the most fundamental level, the universe is irrational, and all we can do is use statistics and probability to say, well, this might happen, that might happen, but there's no such thing as truth. And this became doctrine, which is rammed down the throats of the scientific community, and people like Einstein and Max Planck were pushed to the side, were considered old fuddy-duddies who still believed in the idea that the universe is coherent and rational to the creative human mind. That became outlawed within science on the most fundamental level. Within biology, we had the advent of the doctrine of molecular biology, which developed in the 1920s, which said not that the physical universe is governed and organized according to the same principles that we can know in our own minds, but it all has to be mechanical. This is what became known as reductionism or mechanism, where the cells that make up your body are nothing but little sacks of molecules that bump into each other according to these statistical laws, ultimately governed by a computer tape known as the genes in your chromosomes, which dictate in this same mechanical way every interaction of every atom, of every molecule, and every cell that makes up a human being or any other organism. And that the demand was that every complex process had to be explained from the bottom up in terms of these mechanical, brain-dead laws of probability. And then we had the environmentalist movement came in the 1960s, which said, well, you know, the second law of thermodynamics says everything's running down. Progress is an illusion. Development can only last so long, and then you're going to run out of all the resources that your growth and progress was all based on. So the argument was, we have finite resources. The more we progress, the higher our standard of living, the faster we're going to use it up, and then we all die. So this entropic argument, this finite universe, also became imposed on us. And the very concept of a human being was a little Pac-Man that went around eating up all the food and all the coal and all the resources and leaving behind pollution. So that every new child that was born, particularly in the poorest countries, was just another burden on Mother Earth. And that's what has been left with the young generation today, in terms of a bright, sunny future that we can all look forward to. That's what the scientific method that governed the 20th century has left us with. And if the scientific methods are not changed now, they will kill us just as surely as all the financial and monetary incompetence that created this financial crash. Now, in the case of cancer, it was exactly 40 years ago, 1971, when Richard Nixon signed the National Cancer Act. which promised a war on cancer, that was going to deploy the immense resources of the federal government and the scientific community to win that fight. And while there have been developments in certain areas, in certain types of cancer, overall, the results are still terrible. And I would argue that that is because of the scientific methodology, that we do not approach the question of cancer from the standpoint of what does living, developing biology look like. And Louis Pasteur, the fundamental passion of his research was to improve the lives of people, was to increase the lifespan, to allow people to live full and productive lives, and that the way he would do that was by understanding the unique qualities of living processes where the laws, literally the physics, the physical laws that govern life processes are not the same as dead, inert matter. That there are principles which we see in living systems that don't exist elsewhere. And by looking at those distinctions and exploring those distinctions, we can understand the full nature of the life process, which allowed health, and which would also allow him to gain the insights into disease processes which other people couldn't see, because they didn't know how to look at the problem. And time and again, he was brought in to investigate something that from a technical aspect, he literally knew nothing. Because he had an open mind and an inquisitive mind and a method of approach that would allow him to zero in on the fundamentals that made his discoveries possible. So in the time that we have, we want to give you a brief overview of particularly the early work of Louis Pasteur, where he developed some of these key insights into these unique qualities of life. So, Denise.
You'll excuse me for sitting, but my balance is not well. Okay. I'd like to start off. This photo here is Pasteur very late in his life. I think it's about a year or so before he passed away. And in his arms, he's embracing his grandson. His grandson was named after him. His first name is Pasteur Valery Rideau. And Pasteur Valery Rideau pushed Pasteur's life into the 20th century. He wrote a book about his grandfather and attended many celebrations in honor of the breakthroughs that Pasteur had made. And this, I have a quote here I'd like to read you, and this was on the occasion of the 100th anniversary of the discovery of fermentation in 1957. This is the speech that his grandson gave in honor of Louis Pasteur. He said, "A man of genius was needed to bring light in all this darkness. He was to be Pasteur. This man had the rare gift of insight. You will grant, ladies and gentlemen, that there are two ways for the human mind to gain knowledge: reason and imagination. In the modern world, dominated by technology, we are so accustomed to rational progress that we have come to hold imagination in too small esteem. And yet without it, there could never be great inventors, any more than there could be great writers and great artists. Imagination in the scientific genius assumes a special form of insight. This is the sudden intuition of a truth without the interposition of reasoning. Insight is what makes the scientist of genius foresee the end to be achieved. What contradictory qualities he must possess. Besides the gift of observation, he must be endowed with imagination, so he must be a poet. He must not narrowly be specialized. He must confine himself within the bounds of rigorous experiment, requiring him to bridle his imagination. So, in brief, just brief autobiographical talk on Pasteur. He was born in France in the year 1822, and he died in 1895. I have to say it is a dirty, rotten shame that today when you say Louis Pasteur, 90% of the population says, oh, I know, pasteurization of milk, and has absolutely no idea of the extreme importance of this man's life and what he was able to give to civilization. He, just as in a nutshell, he was able to conquer rabies, anthrax, cholera, and a host of other diseases. He developed vaccines. And yet all he's known for is pasteurization. So we're going to change that. We're going to make Louis Pasteur, well, we're going to remember Jerry by learning the life of Pasteur, because that's what he would like us to do today. So anyway, what happened is Louis Pasteur was working on crystallography. And the reason he chose to work on crystals was because you couldn't see atoms or molecules. And the best way to study atoms and molecules was to be looking at crystalline structure. So you want to show one of those, okay, here's a beautiful example of this. On the right-hand side, you see graphite, and then beneath it are planes where the molecules are attached. On the opposite side is the diamond, and it gives you the boxes, the relationship, the spatial relationship of the molecules in that box. Both graphite and diamonds come from carbon. That's what it is. It's carbon, all the same. Except it's not the same, because graphite is one of the softest things on the face of the earth, while the diamond is exactly the opposite. And it's one of the strongest forces. But this is, you know, to give you just an example of what Pasteur was trying to do. He was trying to get an idea into the unseen processes. He was not a creature of sense certainty. He wanted to be able to understand the way that chemistry and biology worked, as if he were looking into the mind of the creator. You want to give the next one? Okay. This is crystals of quartz. Now, what's interesting about quartz is that, and you'll see this later on when we do some experiments with polarimeter, but quartz is one of the few crystals that has an asymmetric shape. Every other crystal there is that is a non-living crystal possesses a symmetry. This is asymmetric. And you'll find out why, and it's not a living thing, it's just one of these things that pops out in nature, that it's a freak of nature that it possesses this asymmetry. So, Louis Pasteur, show the next one on the crystal cake. Oh, there it is. What you're seeing there is a cave okay, is in Mexico. It's a mile underground. They drain the water out of the cave, and what you see are crystals. And what you see over here are men in the orange. That's how big the crystals got. And they're in like spacesuits because they're a mile underneath, and the atmosphere is extremely hot and humid. That's another view of it. That's crystallography. Now, what Louis Pasteur was interested in is there was a case having to do with wine crystals. You know, you may have seen, you know, on the cork sometimes of the bottle you see little crystals from wine. Well, they're known as tartrate crystals. They occur in all winemaking. And what had happened was, in 1816, a man in Germany named Mitscherlich discovered that there were crystals that looked exactly like the wine crystals had come from a wine process, and in every single way matched the crystals of tartrates, except for one thing. They did not rotate the plane of polarized light. And we're going to get into this later. We're going to do a special demonstration for you. So what Pasteur did is this problem had gone on for decades. And Louis Pasteur, when he was 24 years old, and he was writing his PhD, decided he was going to tackle this problem, which the best minds in all of Europe and the United States couldn't tackle. And what he did was a just beautifully simple experiment. He took the tartrates from wine and he allowed them to dry to form crystals. And yes. He allowed them to form crystals. And on the other side, he took the paratartrates, the other wine crystals that did not rotate polarized light. And he separated, he looked in a microscope and he noticed that these crystals had an interesting structure, that they possessed what they call facets. The left and the right-handed facets, you can see these outlined. This is what he was seeing, well, not exactly, but this is what he saw when he looked under the microscope. So in the regular tartrate acids, he saw all one side of the facet being clipped to the left, so that when it was put into polarized light, it rotated light to the left. He took the second amount of crystals, which no one could explain, and he looked at them under the microscope and he thought, well, maybe these are symmetrical. Maybe they're not asymmetrical like the regular crystals. And he found that they had both left-handed facets and right-handed facets. So this was a fantastic discovery. And he separated out the crystals, and if you can imagine, they were very tiny. And he picked one up after the other, and he put all the left on one side and all the right on the right side. And then he dissolved them again, and voila, the left-handed crystals rotated the light to the left, and the right rotated the light to the right. So this was a major breakthrough, and he was so excited by it that that one of the biggest professors in all of France, a member of the French Academy of Sciences, just fell in love with what he was able to do. What I'd like to show you now is about 13 minutes of a video, a beautiful video that was produced by the young adult movement of the LaRouche organization. It's on the life of Louis Pasteur, and we're going to take the part where he's talking about the crystals and polarized light.
Young Louis Pasteur, at the time a newly admitted student at the École Normale in Paris, had followed these developments carefully. Two acids, totally and completely identical in all of their chemical and physical properties, except that one rotated a plane of light and the other did not. What could account for such a difference? Biot had hypothesized that the cause was in the molecular structure of the acid itself.
This conclusion at the time of its publication especially concerned me. I was at the time a scholar at the École Normale and studying during my spare time the interesting investigations upon the molecular constitution of bodies, and was, as I at least believed, beginning to understand the generally accepted principles of physicists and chemists. Mitselich's communication upset all my ideas. What similarity in all respects? Are there two bodies, the properties of which have been more thoroughly and carefully compared? Can there be, in the present state of science, two bodies so entirely similar without being entirely identical? Mitselish himself tells us what must follow from the similarity. The nature, the number, the arrangement, and the distance of the atoms from one another are the same. In short, this communication of Mitselich's remained in my mind as the chief difficulty in the way of this conception of bodies.
One day, while walking with his best friend, Charles Chapuis, in Paris, Pasteur mentioned his recently sparked interest in tartaric acid and its mysterious twin racemic acid. Chapuis gasped. He could not believe the words that came out of his friend's mouth, for he knew very well the disturbing mystery and tantalizing appeal that these two acids presented to scientists. He refused to let Pasteur get pulled into what had turned into an endless quest for answers for so many, pleading with him to drop the quest and continue with his studies. Pasteur, however, despite Chapuis' efforts, remained enthralled by the paradoxes the acids presented. Pasteur took up a study on all aspects and derivatives of tartaric and racemic acid, provoked to delve even further into the mystery of the tartrates. During one of his experiments, a shocking insight struck Pasteur. All the tartrate crystals were hemihedral, an observation which no one had made before. The young scientist was ecstatic. Here was the clue that would lead to unveiling the mystery of racemic acid. Thus, with Herschel's hypothesis in mind, that there must be an underlying cause for both the hemihedry and the optical rotation in the tartrates, a dissymmetry in the molecular structure, Pasteur followed his intuition.
Mitselish was deceived in affirming that the crystals of the tartrates and the racemates resemble each other. There must be some external differences as regards these hemihedral facets. Mitselish would not have seen these differences, which he did not seek, but I, who have the preconceived idea of their existence, am in a good position to find them if they are there.
Because there was no optical rotation in the racemates, he thought there would consequently be no hemihedry. That is, the molecular structure of the crystals would remain symmetrical. At once, Pasteur replicated Mitselich's experiment and crystallized racemic acid.
But, unexpectedly, the racemic acid salt was also hemihedral. However, the hemihedral faces in the tartrates all lay in the same direction, towards the left. In the racemates, some lay towards the right, and some towards the left. In spite of the completely unexpected character of these results, I, nonetheless, followed my idea. Taking all the racemic crystals, I separated carefully the right hemihedral crystals from the left, and observed the solution of each by itself in the polarimeter. There I saw, with as much surprise as joy, that the solution containing left hemihedral crystals turned the plane of polarization toward the left, and the solution containing right hemihedral crystals turned the plane of polarization toward the right, and that when I took an equal mass of both crystals in solution, the mixed solution, that is half right hemihedral and half left hemihedral, remained inactive toward light, through the mutual compensation of the two equal but opposite rotations. The publication of these facts brought me naturally into communication with Monsieur Biot, who entertained no doubt as to their correctness. Since he wished, however, to discuss these before the Academy, he invited me to come and repeat the different experiments before him. He gave me some racemic acid, which he himself had previously examined and found to be entirely inactive toward polarized light. The solution was then placed in his laboratory and allowed slowly to evaporate. When 30 to 40 grams of crystals had separated, he called me again to the Collège de France to collect and distinguish by their crystallographic character the right and left rotating crystals from one another, under his direct observation. He bade me repeat the declaration that the crystals which I placed at his left hand would rotate the plane of polarization to the left, and the others would rotate it to the right. After this had been done, he declared that he himself would complete the experiment. He prepared the carefully weighed solutions, and when he was ready to make the observations in the polarimeter, he called me again into his laboratory. He first put into the apparatus the most interesting solution, the one which should rotate towards the right. Without making a reading, but upon the instant, he noted a change of color in the two halves of the field of vision. He recognized an important right rotation. Then the excited old man seized my hand and said, "My dear child, my whole life I have loved this science so much that I can hear my heart beat for joy."
Pasteur had found that the racemic acid was in fact an equal mixture of both left-handed and right-handed tartaric acid. This was the first time that the right-handed tartaric molecules were known to exist. Pasteur concluded that any handed molecule which rotates light to one side must always have in potential an oppositely handed pair, though that particular handedness may not be present. Thus the modern concept of handedness or chirality was established. Two absolutely identical but oppositely arranged handed molecules exist for all substances, though, as in the tartaric acid, only one hand may be present. The obvious question here is, why is it in some solutions such as tartaric acid, only one hand is present, while in others such as the racemic acid, both hands are present equally? To answer this question, Pasteur went back to the process which had created this paradox in the first place, fermentation. For thousands of years, human society has used fermentation for winemaking, beer, and food. But nobody really knew what this strange process was. Was it a chemical reaction which caused sweet French grapes to sour and turn into wine? Was it a catalytic factor in the yeast itself, which caused dough to rise and bake into fresh, warm bread? Pasteur hypothesized that perhaps the answer to the curious difference in the tartaric and racemic acids must lie in the same phenomenon that caused the process of fermentation itself. In one experiment he carried out, he fermented a solution of racemic acid. Naturally, at the outset, it was inactive towards polarized light. But as the fermentation continued, he began to see a subtle rotation to the left. This continued to increase as the fermentation proceeded, until the solution reached a maximum left optical rotation, at which point the fermentation stopped abruptly. Pasteur evaporated this final solution and found that only left-handed crystals remained, where there had been an equal amount of both left and right before the fermentation began. The racemic acid had seemingly turned into the tartaric acid. Out of the two possible handed forms which composed the racemic acid, it was as if the yeast, which Pasteur hypothesized was a living process, had literally consumed all of the right-handed tartaric acid and had no appetite for the left-handed form. How was the yeast distinguishing between the two forms of the acid? Did every yeast have its own polarimeter to test the food before eating it? If so, these must be some snobby white-collar yeast. Rather, Pasteur understood that here was perhaps the clearest distinction between life and non-life. Under all non-living conditions, that is, without a process of fermentation acting on the racemic acid, the left-handed and right-handed tartaric acids behave identically in all respects, except for the direction in which they rotate light. However, to a living process such as fermenting yeast, the two forms are as different as night and day, to the point that the yeast chooses only the right-handed form, ignoring the left-handed form completely. Therefore, tartaric acid would only appear in, and was only a product of, living processes.
Sure, you understand a little bit more what I've been trying to explain about the tartrates, but we're going to go even further, because I'm going to have my husband come up and actually demonstrate this on this little polarimeter that he built for me on my birthday 7 years ago. I started my quest of examining Pasteur and his life back in the year 2004. And I was completely excited about this idea of a polarimeter. How does this work? So I went online and I Googled up polarimeters, and I found out that they cost $45,000. And I asked him for one for my birthday. But instead, what he did is he turned around and he made me one. This is, in a sense, the same process they made polarimeters back then. And I'm going to let him talk to you and tell you what he did.
Yeah.
I'm sorry, before you ask another question, as a teacher, maybe not a very good one from Wednesday.
Please.
Sometimes our ego gets in the way of learning things or asking questions that enable us to really understand things. So the very basic idea of rotating light, could you just explain exactly what that is?
That's what he's going to do.
In fact, I think I can probably do it on the screen, hopefully. Everybody's had polarized sunglasses at one point or another, I imagine. This is a pair, an old pair that I took apart. If you just look through polarized sunglasses, it just tends to make everything dark. But the reason that they are effective to eliminate glare is that usually glare is bouncing off, excuse me, could I get some water? Is either bouncing off the surface of water the surface of water or maybe the hood of your car, and in the process the light is only bouncing in one plane, so that the reflected glare is light which is moving, let's say it's only moving vertically or it's only moving horizontally, the actual oscillation of the wave of light. So if the wave of light is moving this way as it bounces off the hood of your car, if I were to put a picket fence in front of that, the wave that's oscillating side to side cannot get through. Whereas a wave of light oscillating vertically can go right through the boards. That's the principle of a polarized lens, which you normally don't see if you just are looking through it, because light tends to reflect in all different axes. But what you can see, let's see if this works. If I hold up one lens, it just makes the screen dark. If I rotate it, you can see there's a difference in the color. If I put one on top of the other in the same orientation, it's darker. But here's the real kicker. If I rotate one relative to the other, it becomes completely black. That no light can get through the two lenses because one is oriented vertically and the other's oriented horizontally, so that the amount of light that passes through will go from a minimum to a maximum. Okay, can people see that difference? Okay, so the idea of a polarimeter is, first of all, you have a lens in front which light will pass through, which means that only light oscillating in a vertical plane is going to go through the tube where you have a sample of some material. And the question that you're asking, does the material that you've put in here have any effect on this polarized light? And if you think of it, you're taking a form of light which has a very specific property, and you're asking whether the substance has any effect on that light. And this originally came up with quartz crystals and other things where reflected light seemed to be altered in some way, which is what sort of led to this idea that you could have polarized light and could use it, again, as a way of trying to see something in the structure of materials that you could not see. To see the unseen, just like seeing the atoms or the molecules. So the idea of the polarimeter is you have two lenses, one which comes before your sample and another one after the sample. And when you look through both of them, you can align the two lenses without any substance in between, so no light passes through. And then you put a substance in between the two lenses, and what you may see is that now, to get that complete minimum transmission, now instead of having them perpendicular, one has to be rotated 5 degrees, 10 degrees, 20 degrees, until you again are blocking out all of the light. Is that clear so far?
So it's sort of counterintuitive in the sense that you put something else there, and it's not as easy to, or it's not as direct. It's not as direct an understanding of what you do to block out light as it was before. Is that?
Well, the interpretation would be that somehow between one lens and the other, the substance is rotating this plane of polarized light, which you can detect by this second lens. Okay? That's the basic principle. And what I'm going to do quickly is, this is the polarimeter which I built, which has a little candle in the back. This is just a tube. There's a polarized lens attached to the front of this, and another one here. And in between, we can put a sample of something. And so if I first align these so that they're perpendicular, which I measure by reducing the light I can see from the candle to a minimum, and then if I put something in between, I'll see if that changes. And then I have a dial on the front where I can rotate this to see if there's been any effect. Okay? So that's the hypothesis. Now what I want to do, Marcel, you want to come up since you're have to have a guinea pig. What did I do with the? Oh, we need a chair. If you here, we can. If you want to. Okay, so sit down in front, and focus. Okay. So what I want to do first. This is set on 0. So now I want you to adjust the back, this back tube. You can just go back and forth until the light disappears or gets as dim as possible. I don't think you need to.
I don't think you need to do that.
If you go, when you go back and forth, you'll see the light will get bright and dim, and you can sort of zero in on that.
I think at this point here, the light is about as dim as...
Yeah, it doesn't completely disappear.
Sure.
But it gets very dim.
Right.
Okay. Now, what I want to do, I just need to slide this back a little bit. Okay, is that still the place where you want it?
It's still roughly as dim as it was.
Okay. Now, this is a jar that just has tap water. So I'm going to put this in between the path here. Make sure we got enough room.
It's blocked it out almost completely. Even more so.
Okay, but it's just tap water. Let's make sure this is got enough room. Okay. So if you want to adjust it again, this would be sort of our control. If it's just tap water, we don't really want to measure. Maybe water does something, but probably not.
You need to make it as dim,
As possible, yeah.
Okay.
Just sort of readjust it.
It's still very dim. What is that the dimmest?
Yeah.
You get dim light because the wavelength is not perfectly in one axis. In other words, to make the candle source, they supply slightly variations. Is that right?
Well, it's going to vibrate in a lot of different directions. The idea is the first lens only lets this light through, and the second lens only lets this light get through. So between the two lenses, not much of anything gets through. Okay? So by looking for this minimum transmission, we're aligning our two lenses like this. Okay?
So now the band where it's at its dimmest is a lot briefer. In other words, there's not a larger degree of rotation where it remains dim, but I think
it's just sharp. Okay. Now, this is a solution of glucose, which I got from Caro syrup, I think. This is sugar, which is the product of what?
Life.
Of life. Okay? So, what was being investigated in the video was most molecules, just because they're complex and they're three-dimensional, are going to tend to be asymmetrical. There's going to be something sticking out on one end that's not on the other end, so it's asymmetrical. And if you were to produce that in a test tube in a laboratory, there would be no particular reason why you would get one mirror image or the other. This is why they call it handedness, right? If you look at your hands, they all presumably have the same number of fingers, and there's a thumb on the side, but you cannot superimpose them. If you superimpose them, well, now I'm looking at the palm of this hand and the back of this hand. So they're symmetrical, but different. So if you're making something in a test tube, you would get half of this and half of that. That's what they found in what they call the racemic acid, which was not affected, which did not affect polarized light. But what Pasteur discovered was that chemicals that were the product of living systems, like sugar, that you wouldn't get both forms. You'd only get one. And the way that you could find that out was by putting that sugar in our polarimeter. Okay, now I want you to turn this front lens, which is calibrated, to see if that dimmest spot where it cancels everything out is the same as with water, or if it's been rotated. So just turn the thing with the needle on it, that front part. So go back and forth and see what's the dimmest place.
Well, looks like it's got a redder glitter.
Okay.
Right? Where it's dimmest.
But try to find its dimmest. Just sort of go back and forth a little bit until you can zero in on it.
Very narrow band, but I would say at about, what is it, 20 degrees right.
Okay. So if the first lens was like this, and the solution rotated the plane of the light that came through the first lens, now to get that same minimum transmission, I have to rotate the front lens by that 20 degrees. And I'll just, thank you Marcel, you were an excellent subject. You got the result I was looking for. And I need one more volunteer. I need one more volunteer.
Who wants to see how this works?
All right, come on up. Okay, we're going to do the same thing with a different solution of sugar. So first I want you to rotate this tube. I just put the plain water back in again. So we just want to, this is sort of our standardizing it. So grab this tube and rotate it back and forth until you've got it at the least amount of light coming through. Okay, you happy with that? But it's as small as possible. Okay. So we're going to take the water out. And this is also glucose, but it's a stronger solution. So there's more sugar in that sample of water.
Now that's yellow.
Okay. Now I want you to turn this disc with a needle on it. Not that, but this front lens. Rotate that until it looks as dark as possible. Does it look about as dark as possible? Okay, and what does that say? Okay, 35 degrees. So both of them rotated the light to the right, one by 22, the other by 35 degrees. So the amount of material that somehow, and get this has always perplexed me still. This is a solution. So whatever the nature of the structure of the sugar molecule is, some are like this, some are upside down, they're moving around, yet the cumulative effect of that, even in a solution, is to rotate it consistently in one direction or the other. And Pasteur's insight was that this tells me something about what made this. That living processes can be detected. I mean, this is one way that you can detect products of biological processes that don't occur in a chemistry lab in a test tube. And that what we found is that every amino acid, every protein, every hormone, just about everything you can think of that can have this dissymmetrical structure, you will find in living systems only one of the two. So this is a perfect example that living processes don't act like billiard balls bouncing into each other.
Repudiates the chaos theory.
That's right. So this was one of the early discoveries. He's a kid. Did we put up the, yeah.
Yeah.
Was he 23?
24.
24, he made this discovery, which rocked the scientific community because this had been a huge deal. At that time in the history of chemistry, which is what Pasteur was trained in, he was not a biologist, he was not a medical doctor, yet he made major contributions in those areas, much to the chagrin of all the PhDs and MDs. He recognized that this was a qualitative difference, and that was important in everything else that he did. So, you want to?
Sure, just thank you.
Okay. We'll have this if we have any time.
If we have any time, we can have more people come up.
We're running late now.
Okay. Now we've examined that. Go back a minute, and right at the end of that youth video, you saw that he was looking into fermentation. What happened was he became the top professor at the age of like 28 at the University of Lille, which was the key industrial center of all of France. And in Lille, he had these classes where his students would come in and he'd lecture some, but he'd actually push these kids to go to the lab. He wanted them to have hands-on experiments in the laboratory. And then once a week, he opened up his class, I think it was on a Saturday, to anybody who wanted to come. So all of the industrialists throughout France got very excited, and they decided, because they heard from their kids who had studied under Pasteur, so they came and they would sit. And one day there was a young man.
Can we just show a couple of these pictures?
Oh yeah, sure. Go ahead.
Before we get to fermentation.
That's fermented. Okay.
That's tartaric acid crystals. Go ahead. This is just a picture again of these, the right and left-handed. You can do this with styrofoam balls and toothpicks, but you can construct molecules that have exactly the same chemical formula, that have exactly the same units that comprise it, but have this dissymmetrical orientation, that there's no way that I can take this one and turn it around and make it look like that one. And just one thing about this is that Lipitor, naproxen, ibuprofen, you know, most anything you can think of, these come in these symmetrical forms. And one of the most dramatic examples of this is a thalidomide baby. People remember back in the 50s and 60s, pregnant women who were having morning sickness were prescribed thalidomide, which is both sort of a sedative and to help settle their stomachs. And they found that they produced these horrible birth defects of deformed or missing limbs. Thalidomide comes in two forms, one of which cures morning sickness, and the mirror image causes these birth defects. So there are some things, I think it's ibuprofen, I forget which one it is. There's ibuprofen, one form cures your headache, the other form does nothing. So they don't bother to separate out the mixture or to worry about that. But naproxen, one of the other pain relievers, one cures your headache and the other is toxic to your liver. So when they manufacture it, they have to figure out how to produce only one form and not the other. Okay, whatever. I've forgotten the specifics of it, but so this has become a multi-billion-dollar industry in the pharmaceutical industry to figure out ways in a sort of chemistry lab setting to do what biological systems do naturally, to produce only the desired form of these complex molecules. So, okay, that's all I wanted to add.
Want to go to the next one? Okay. So, a man named Bigot, who had a big beet juice factory, they made sugar, came to Pasteur and said, it's growing rancid. Why is this happening? Can you tell me why? And Pasteur said, well, I'll bring my microscope. And it was the first time that anybody had ever brought a microscope to a manufacturing plant. And what Pasteur did is he asked, what is the process by which you produce this beetroot juice? And he always did this. Every time he went into a foray into, you know, a new area, he would always pick the brain of the person who knew something about it so that he would become an expert in it as well. So he picked his brain and he sat down with his microscope and he began to see these globulars, and that what was happening is that they were moving and they were dividing and it's a cellular concept whereby they were multiplying. And he recognized immediately that this was a life process. And up to this time, it was believed that fermentation was merely a chemical reaction, where Pasteur said, no, this is a process of life. And that was the first time, and there were a lot of people who didn't believe him. I can tell you right now, hosts of people who did not believe him. But this is what he proved. And
that's yeast.
That's yeast. I didn't say that. Okay, this is yeast. You want to show the next one? There's another yeast bunny. The next one. Okay. Now, because we're running out of time, I'm going to go through this as quickly as I can. But Pasteur took on a 6-year project in between everything else he was doing. And he was refuting a doctrine known as spontaneous generation. Spontaneous generation says that life comes from non-life. And this was something that Pasteur knew could not be true, but he had to prove it. And everybody who was around him, the top scientists, said, don't tackle this, Louis, because what's going to happen is you cannot prove that something that does not exist, it's too hard, don't waste your time on it. And he said no. And he was pitted up against another scientist named Pouchet, who claimed that life came from non-life. And what Pouchet did is he would collect dust from all over the world, from temples and, you know, churches, and he would put the dust and whatnot in water, and pretty soon you'd have germs. And he'd say, see, this arose miraculously out of the water. From dust, these germs appeared.
Nice.
Oh yeah, and then it was also commonly believed in the Middle Ages that if you put a dirty rag in a corner with some corn, that mice would appear. And they would mysteriously grow out of the dirty rag and the corn.
Yeah. It was the middle 1800s.
Yes, this was accepted up until that time. There was no concept of, you know, sterilization, cleanliness, germ theory. This is the beginning of germ theory. What Pasteur did is he wanted to prove that life came from life. He invented this flask here. It's called the swan neck flask. And what he did is he took sugar, yeast, and water, and he boiled it until all germs were killed. He had it in this form in here, and he put a little cotton wad at the end of the tip there so air could come in, but it would keep the dust and whatnot out. And this would remain perfectly sterile, perfectly sterile for as long as you wanted, unless you broke the neck. If you broke the neck, the germs would go in, and voila, you would have, you know, germ-laden material. And Pouchet, he did not believe this at all. What Pouchet said is, Pasteur, you're saying germs are everywhere. If they're everywhere, then there would be so many germs that the air would be as dense as iron. We couldn't see. So anyway, what Pouchet did is he went to the mountaintops and he took hay and he cooked up the hay in the water and brought it back down from the clear mountain air, and all of a sudden germs started to appear. And he said, see, this came from water, you know, from the pure mountain air, and germs appeared out of nothing. And Pasteur said, my friend, you boiled the hay, but not long enough. You didn't kill the spores. So there was a big, finally the whole thing ended with Pasteur coming in with 60 of these, and one after the other he would demonstrate breaking the necks, and the guy gave up after 20 and left the room. So anyway, that's the end of the formal presentation. What we have up here, if you're interested, are products of of living material, and you'll see this idea of asymmetry in these things. We have crystals, we have the polarimeter, and we have a few minutes, so if people would like to come up and see this or work the polarimeter, I invite you to do so.
Just the idea, when light goes through any kind of a substance, the molecules, so the molecules are screening or deflecting, or how is that?
They're causing a rotation of the plane of the light. Exactly how that.
So there's interference in certain planes, so the light won't go through that, but where it's allowed to, it goes through. So each one has its own characteristics apparently on that change, you're saying.
Yeah.
And if it was balanced, it would kind of like neutral out. So,
Yeah.
You know,
That was what they found in this so-called racemic acid, which did have crystals. When he actually dried out this solution of racemic acid, he found tiny little crystals of both the left and right-handed form. And if you separated them, the left-handed would rotate light to the left, the right-handed would rotate light to the right, so they were optically active. But because they existed in a 50-50 proportion, that when both crystals were dissolved, it had no effect on the polarized light. So it wasn't that the racemic acid actually had a different chemical structure. It was purely this distinction that a living process, which is where these tartaric acids would normally be found in wine vats as a result of the fermentation process, that only one of the two isomers would be present. And that's why they rotated light